This RI Hubs project. Oh yeah, so let me just say one thing that Yara told me. So we are going to record the presentations by Adan Guerrero and Katy Wolstencroft. But after that, we'll have these two presentations, and we will try and have all the questions and answers and comments after, once we have all the information. And recording will be stopped at that point to really discuss freely and be quite flexible in that part. It's going to be really important to try and don't be shy and discuss, bring up your own comments and questions. So actually, this project is a 3 years project that is connecting 6 different countries, 3 in each region: Latin America and the European Union. So we have Italy, Spain, and Portugal connecting to Brazil, Uruguay, and Peru. And the whole idea came from the European Union, Latin America, Caribbean interest group, which is working for several years now in trying to improve the cooperation between the 2 regions and in terms of research, and in particular, in terms of research infrastructure in this case. So 8 full partners, 2 European research infrastructure consortia are working in this project as associated partners, which are Instruct in structural biology and Euro-BioImaging in bioimaging. And we have also 2 Latin American networks in these areas called Lobby and SEBEN. So let's think each one of the national agencies that are actually giving the funds for this project to work. So this is both interesting because of the actual cooperation, but also complicated to bring about all these different agencies and make the project move smoothly forward. So just a word, this idea of hubs focused, of course, in our case, in multi-scale bioimaging. By multi-scale, I'm meaning going all the way from, photonic microscopies to see whole organisms and tissues and cells, all the way to atomic resolution techniques to image molecules that are important for biology. So this multi-scale bioimaging infrastructure, you know that you can classify different ways, different flavors, but there are large facilities and entry-level facilities, and these both play really important roles that are not substituted one by the other, right? So the whole idea of this project is about providing the glue between these types of large, high-end instrumentation and entry-level facilities that would provide easier access to a broader community of users. And we want to do this together between 2 different regions, as are Latin America and Europe, because we do believe that by developing these hubs that will kind of funnel in a larger platform of users, we could improve the coordination of activities amongst us and really make this talk between the different scales of facilities much smoother and effective, right? We want to establish common procedures to run these infrastructures, and particularly pinpointing and solving, hopefully, issues that concern the exchange of samples, the exchange of data, and the exchange of people. 3 axes that we're really working on actively. And the last, not least, is to focus our capacity building in the ways we need. So via a couple of initiatives that we're carrying on through RI Hubs, which is the staff exchanges that is happening, and training activities. And this very first webinar is actually launching one of the training activities that we conceived. And this is thanks to the work of the training committee in our consortium. And I want to name people. I want to thank Karina Alieva, who is now here. She's chairing this committee from the Centro de Biología Estructural del Mercosur in Buenos Aires. Also, Claudia from Instruct, who she was the first chair and kicked off the whole organization of the committee and put it in motion. Thanks to Yara, Oliver, Andrés Camay, Rodrigo Portugal, and also Oscar and Blanca from the Centro Nacional de Biotecnología in Madrid. All of these guys are the ones that make this happen. So one of the first things was making a survey, an internal survey, to just pinpoint topics that would be needed by the the different nodes of the consortium, and actually management of research infrastructures came on top. We're not talking about that today, but it's going to be definitely one of the important things to work on and train our people in to professionalize that. But data management came also very high. How to manage, how to organize, store, share, and so on. And this is a whole field of work. And we have the honor to have Adan Guerrero and Katy Wolstencroft to talk to us about this today. So just the end of it is, this is pre-RI hubs, the interactions between Latin America on the left, Europe on the right, amongst these people working in bioimaging. And this is what we want to generate, like this kind of web. And it is actually happening. So many of these, or a few of these new arrows are actually there, and we hope to increase this by opening it to the whole communities. So please, be in touch, and we want to be open to the communities in both ways. So with that, let me stop sharing myself and invite Katy Wolstencroft, who is an assistant professor and head of the Life Science Semantics Group at the Leiden Institute of Advanced Computer Science in Leiden University in the Netherlands. And she has published numerous articles that reflect her interests and seminal contributions in the areas of semantic data and knowledge integration for biomedical data science. So this is really interesting for all of us. She has given a particular focus on fair data. She will probably talk about that, and reproducible research. She was actually directly involved in defining the FAIR principles: findable, accessible, interoperable, and reusable principles. So thank you very much, Katy, for this. And the floor is yours. The virtual floor is yours.
Thank you very much for a very nice introduction, and also for the invitation to speak today. I think this sounds like a very exciting initiative, so it's really nice to be part of the beginning of it. I'm going to talk to you today largely about FAIR data and how we're using the FAIR principles to help us build infrastructure for bioimaging in the Netherlands. I lead the data and analysis components of this national initiative. It's quite a new national initiative. My slides are not working. Yes. So we started actually last December, so we're not even a year old yet. It's a 10-year project, but 5 years fully funded. And what we're trying to do is to elevate bioimaging across the Netherlands by making more resources available to all researchers. And those resources are different kinds of microscopes to enable microscopy at various biological and technical scales, to enable better, newer AI-driven techniques on the image analysis, and also to make better use of the data. So I will be focusing on that part today. As has already been said, I was very much involved with developing the FAIR principles. I am not a bioimager. My background is in bioinformatics. So I came to this with all of the data integration problems that I tackled in bioinformatics and also systems biology, where data integration is one of the main concerns. But actually things are not very different. I think in bioimaging, we have to not just think about publishing data in a fair way, but supporting the scientists in making better data management and fair choices all the way through their research. And so we wanted to design an infrastructure to enable support for scientists through the whole data lifecycle. And this is what you can see here. And that means actually building on what has gone before, not reinventing things, but trying to formalize and standardize what the community has already started to do anyway. So at the beginning, we started by thinking about all of the resources we could use. There are very generic data resources like iRods. It's very popular in the Netherlands. But for bioimaging, of course, Omiro is already some kind of de facto standard for managing data. Lots of instances of Omiro are instantiated around the world. So this is the way that we decided to go. And of course, there are advantages because the IDR is also an Omiro instance. Similarly, there are various standards and ontologies that we can make use of, and there are various analysis tools that already plug into Omiro. So we're building on top of community decided standards and best practices. But what we actually have to do is tackle the distributed nature of our instruments and our people and our resources in some kind of tractable way. So we have 18 different institutes. They're all generating data. We have to standardize the way that we collect and organize the data at each node, even though each node has different resources for doing that. So we need to make it as easy as possible. So we have now come to a position where we've proposed a kind of minimum standard for data management for the NL bioimaging network. And that is based on, I think everybody's seen the Omiro database before, but if not, this is a screenshot of the Omiro database. And you can see in the middle thumbnails of various images, and then places for metadata on the right, and folder structures showing how data is organized within the database. But what we want to do with our infrastructure is build on top of Omiro to improve the fairness, to improve the support for the rest of the data lifecycle. And so we did that by instantiating Omiro at a few sites initially, and learning how our users work and building on top of that to help improve incremental fairness throughout the process. So we started looking at support for Omiro with just a few microscopes in our facility to begin with, 4 microscopes, 60 active users. We did this during the pandemic. So actually we had lots of motivated people who wanted to join in because they didn't have access to their microscopes. They were stuck at home. They wanted to better use their data that they already had. And so this was in a way, it kind of worked out well for us, that whole period of staying at home. But by doing that, we were able to instantiate some good practices that were useful to our community. And then we rolled these out in full in February 2021. And now actually Omiro is connected to all of the microscopes in the facility, and we have a very large number of active users. And this is the way that we collect our data and metadata now. And this is the way that we interlink with data analysis. And of course, this doesn't happen all by itself. We have to help the users get used to the system. We have to provide training, documentation, and support. So we have a yearly microscopy course. Everybody who's new is given an onboarding training. We have the promotion of Omiro for use in publications. We spend a lot of time talking about different data management solutions and how actually researchers now need to come to some kind of, researchers are required to keep their data for 10 years within Leiden. So by doing this the way that we recommend, it's cheaper, it's safer, and it's more user-friendly. The PIs know where all of their students' data is. And so from a local point of view, this is already showing to work very well for our community. And of course, there are already good ways of interlinking with analysis tools from Omiro, but we've been working on improving that as well. So this year, this is what we've been focusing on. We've been instantiating this solution at various other places in the Netherlands and working on how different researchers across the Netherlands can access the data in these resources, and different researchers outside of the Netherlands as well. And we've been making it easier to roll this out further by building Docker images and these kinds of features. But that's just at the local level. What about sharing further afield? Well, we want to store and share data efficiently at a local level while people are working on it. But of course, eventually we want that data to go into the public repositories. And so we need to help users migrate their data to the public repositories. And that means working on the standardization of the metadata so that process can go smoothly. Because the data is local, we want to still be able to disseminate the work of the Netherlands in general in bioimaging. And so we're also building a national FAIR Omiro node. And this is just for metadata sharing. It's not for data sharing. The data stays local until it's published, but we can at least tell each other what we're doing. The people in the experiments are just as much research assets as the data and the analysis tools that are being used. And so this is where we are now. In terms of the national infrastructure, we have a few Omiro nodes established. We're working on this national metadata sharing node. But at the local level, in order to get the data from local Omiros to the image data repository, for example, we need to work on standardization. So we've been trying to help our users standardize their metadata, package their data, metadata, and analysis tools in fairer ways to make that whole process more accessible. And again, we've been building on resources that are already available. REMBI came out in 2021, recommended metadata for biological images. So this is a standard that we have all, as a community, decided to make use of. This is a general standard for all microscopy, but actually we also want to link to other kinds of data. So we needed to think about that as well. There's another standard that would help us do that, investigation studies and assays. And this is a very simple way of aggregating your experiments to say these assays belong to this study. This study shares the same biological samples, for example, and they all belong to this investigation. And so what we've been doing in Leiden is consolidating these standards so we can actually say how ISA and REMBI fit together. And instead of trying to do that for the whole of microscopy at once, we've been focusing on what we specialize in Leiden, which is high content screening. So we've developed a REMBI and ISA compliant metadata model for high content screening. It's called MixMe. And we've been following the same procedure for other kinds of microscopy that is common in Leiden. And other places in the Netherlands network are following the same methodology to do this more broadly. So now it's become a method of ensuring that we have standardized metadata in a very user-friendly way. If you're interested yourself in using these metadata templates, they are published. Well, there's a paper about them, but the actual metadata templates are also published. It's all fair and open. So we have the templates themselves, and we have 4 examples of data sets that have been annotated using these templates. And we've shared them in the FAIRdom Hub. And the FAIRdom Hub is a project commons, a FAIR project commons that actually I developed together with a different consortia from a previous project. And there's a public version of the FAIRdom Hub available for anybody to use. And so we're using this to store metadata templates. We're also using the FAIRdom Hub as a metadata sharing framework for the national node in NL bioimaging, as I mentioned before. And by doing this, we will be able to not only link our microscopy data together, but also interlink the microscopy data with other kinds of omics data, but just on the metadata level. It doesn't matter where the data is. It could be stored within the national node, but it could also point to local databases in other places or public databases or various different combinations of those things. The FERDM Hub is probably a topic of a completely different presentation, but this is the basis for this national metadata sharing node. And the idea there is that we can better integrate with resources at the European and global level as well. And so we've been quite busy in our first year. I think what we're trying to do is to support the users end to end, from the laboratory experiments and the laboratory metadata to all the way through to analyzing the imaging data. And I've spoken mostly about the data management part, but of course we're also focusing on the image analysis part. And we have been developing a containerized system for storing and sharing bioimage analyses. And we have now a plugin for Omero called BioMero, which enables you to run these analyses more easily on high-performance compute facilities. The paper's just come out. I think this is the archive version of the paper, but there is actually, the paper's been published now. I should have changed that link. But the idea here is to make a uniform way of providing these kinds of analysis service. And so users can make use of collections of these analyses, and they can run them on high performance compute that is local to them, or it could be somewhere on the cloud. It really is quite flexible in that regard. So I'm going to draw to a close now so that there's plenty of time for discussion and our other speaker. But I wanted to give you a flavor of all the things that we are doing to enable nationwide fair data sharing in microscopy. And over the coming years, we're going to be extending and expanding this idea to other kinds of microscopy, to the rest of the nodes in the network, and also to better interlink with the resources from Euro-BioImaging and global bioimaging. Thank you all for your attention. There's a lot of people to thank, the national team, the Leiden team, and many other people who've been involved in the FAIR data initiatives as well.
Thank you so much, Katy. It was really, really nice. And indeed, what we're going to do is listen now to Adan Guerrero's talk first, and then we can really open the floor for discussing. I have several questions for Katy myself, so I want to introduce now Dr. Adan Guerrero, who's a research scientist working at the National Laboratory of Advanced Microscopy of the Universidad Nacional Autónoma de México, the university in the city of Mexico. His work focuses on the development and application of bioimaging technologies to understand nature at micro and nano scales. So making him a pioneer in super resolution microscopy. I think it's going to be quite complementary with Katy's presentation. Adan has played a key role in fostering the growth of the Mexican bioimaging community and actively participates in several of these networks that Katy mentioned, like Global Bioimaging, LABI in Latin America, Bioimaging North America, and so on. So he's very active and has received many awards, including a prestigious Young Scientist Research Award that the Academy of Sciences in Mexico granted him in last year. So thank you very much, Adan. If you can share your presentation, and please.
Thank you so much. Now I'm sharing my presentation. Let's see if you can see it. Yep, this share. Okay. Can you see my presentation there?
Excellent. Yes.
Right. Well, thank you for the invitations. As you mentioned, I'm participating in these global efforts to foster the development of bioimaging across all the globe. And I work in this laboratory, which is a network of advanced microscopy, advanced bioimaging microscopy in Mexico. And we are really pushing forward to change that name to growing the Mexican bioimaging community. So last year we had the opportunity to organize the Labi ex meeting, which happened in Mexico. And in this meeting, we were mapping some of the initiatives that are happening across America. So if you want to know more about this, we wrote this paper, which is called Building Momentum Through Networks by Imaging Across the Americas. So what we put in that paper, and I'm sharing you now some of the images of the paper. Sorry for this DUI that appeared just right there. So I'm just taking it out. Okay, so what you can see is that there are several places in Latin America that can be or cannot be considered as bioimaging hubs, depending on the activities that they are developing in their communities. That can be like providing open access infrastructure to the people, to being key, to having key role in the development of bioimaging in their countries. So we have mapped several of them all across America and Latin America. And so if you want to see some of these initiatives that are actually running that complement with some of the initiatives that are presented in this paper, please go and see this paper. So in addition, we mapped what are the programs that foster the education of bioimaging in Latin America that go from postgraduate programs that are specialized in electron microscopy technologies, artificial intelligence, amongst others, and also training opportunities that happening all around the America. But to be part of this presentation, so in the meeting, we also started discussing the problem of handling image data science, and specifically what is the mapping that scientists have to follow to go through this ecosystem. So starting from laboratory experiments, with information about the acquisition system, how to integrate information, how to visualize, analyze, publish, and all the going and forward between analysis. And so this is a great scheme that I like it, which is what are the specific tools that we can find in each of these steps, which is called the bioimage town, where you can find some of these image data management system, which is a matter that is actually really well adopted by the community all over the globe, and also other applications to handling the data, like specific formats to move the data, for example, in the web, or to move the data to high content image analysis platform. So this is a really complex metro station that we always have to bring in consideration when we are aiming to develop an imaging experiment in this era. And for me, and I think that for plenty of people in Latin America, is how to become knowledgeable about the intricacies of this ecosystem and avoid getting lost within the metro map. And that is a huge question, because there are several technologies that are arising, and some of them will last, some other not. So I start asking to ChatGPT and ask it to look into the internet. So ChatGPT gave me this solution. To avoid getting lost in this complexity, I suggest you the following. Define your scope. So determine what are your specific needs within the bioimage data management. And this has to be considered for the people that is starting an experiment, or for people that is handling a facility, for students, for PIs, for all the people involved in this ecosystem. So, and also identify the key areas that require focusing, like data acquisition, storage, processing, analysis, and sharing. So it's important to map this complexity, and it's important to map this complexity from the beginning, and also it has to be coordinated. It doesn't need to be an effort of a single person. So, and if we think about data, so data is multidimensional, as you already know. And when we are generating the data, it's not only about the data, but also about where we can get more meaning about the data, which is the metadata, and how we can assess quality of this. So data management and sharing is essential for this. So in this case, metadata is important for interpret, reproduce, and share the information, but also we need ontology, which is about the design of experiments that was happening with that data and related to the specific intricacies of the data, like phenotypes of the cell lines or the organisms that are deduced. And also is related to how the information is captured, is stored. And of course, not always all of this metadata is accessible. So it's important to be aware that this ecosystem is growing, and there is plenty work that is being done to make it more accessible and to make it more transparent. Okay, so at the end, what we need is data management. And so I took these slides from the Bioimaging North America meeting. So those are slides for the corresponding working group of metadata and data management and sharing of BINA. And what we have with the data is an elephant in the room that we all know that it is there, but we need to really know how to handle this complexity. So, and for that, also ChatGPT recommended to understand the FAIR principles, familiarize yourself with these ones that are the way in which data has to have unique identifiers. It has to have compatible with other databases, has to be open, free, universal, and clear and accessible, and I mean, it has to be reusable. The end. So, and what is actually happening is that the ecosystem is growing at all scales. And for example, in the publication ecosystem, I recommend when people is starting new experiments to go and look at the guidelines that are in the editorials, because there is guidelines there that people can follow about reporting standards, by availability of data, materials, scopes, protocols. But also we are on the step that we are pushing editorials to put more standards about image integrity, and also related to the acquisition of data in microscopy. So I recommend you to go and see before publishing, before designing the papers, to check what are the global needs that is being pushed out of these groups in which we all participate. So another recommendation that was given by ChatGPT is understand what is out there related to data repositories, and here by the Bioimage Archive, the Image Data Resource, Empire, which is for electron microscopy, and others came out. And I will also include other databases that are actually gaining momentum, like the System Science Biological Dynamics database, which is in Japan, and Nacional, which is more open to other ecosystems, where you can put not only microscopy but also papers or genomes, or I mean other sorts of information. So these platforms are being built to create an ecosystem where data can be accessible, organized, stored, and interoperable. So we also have been speaking about how to handle the metadata, and what are the standards that are out there to handle the metadata. And there is plenty of work working with companies also to define and to create the standards that are accessible for all the people. So there you have some of the QRs for some of the databases that we are speaking about. But what I want to give you is some examples of value about using these databases. And I will take the concept of reusability. So for example, in this case, so we published a paper in 2022. And what we did in this paper is that we put all the data accessible for the people, where the readers can download the raw data. The raw data is already in Zenodo. And also we connect that raw data with the scripts for the analysis for the software, which is also connected through Zenodo. So the ecosystem is being built. It will be asked by editorials, but if not, I really recommend you to teach people on navigating this ecosystem, because it's pretty new for most of the scientists right now. So this is another example that we are building here in Mexico to show what else we can do with the data, which is reusability. For example, what if we just download the data, which is in the databases, analyze and get more meaning out of the data. For example, in this case, we download images of image-based flow cytometry, and we process it with algorithms that increase resolution of the images, and then we can extract more information, which is more about the local intricacies of the information in the cells, so which can be quantified and can lead to novel discoveries. So I also encouraged you to encourage people to reuse the data that is out there. It's not only to store the data and to make the publication, but also to use it even more. Okay, another recommendation that I got from ChatGPT is engagement with communities. So there are plenty of communities there, like Global Bioimaging and Latin American Bioimaging. Of course, communities are growing, changing their names. So NEBIAS, which was an European network for bioimaging analysts, can change name to the Global Network, and so on and so forth. But it's important because all of these organizations provide resources, guidelines, and expertise that can help the people that is aiming to enroll in this complex ecosystem. And the most important thing, I recommend people to engage more in forums like this, where they can learn about what are these complexities. So at the end, so for example, this is one of the working groups which is running in Bioimaging North America, which I'm participating, which is the quality control and data management. And in all of these groups, we are trying to addressing this elephant in the room problem, which is going to standardize aspects and to learn how to manage the data, and also to provide the standards for the global community. So at the end, we try to work out together, for example, in this with Guerrero and other organizations that are really taking the lead on hoping for a more fair world in terms of data. And also a very important aspect is training and education, as awareness is one of the major things that we have identified in Latin America. And for that, for example, in the previous Latin American Bioimaging meeting, which happened in Rio de Janeiro this year, so we had a specific session dedicated to data management in biomedical. In this meeting, we launched the working group Image Data Science of LABI, and we addressed some of the we interviewed the participants for what are the problems that they identify in their communities. So, for example, one problem that we all identified is funding, but also data management. Data management is right now a huge problem in Latin America, and it is very difficult to keep track of the data that is being generated within the facilities. So there is not knowledge about the standards, the guidelines to follow, how to store and handling the data. And so there is also a need for more image analysts that help the community to explore the data that they are generating. And of course, so that there is nowadays plenty opportunities for technical training, but little opportunities for training or for analysis or data management training. So in some countries, so this is a really important observation, that is in some countries in Latin America, it seems that the general concept of data management is not present at all. So this is something that we have to build. And also the access to infrastructure is limited, and so on and so forth. So at the end, what we have seen in the previous presentation is accompanying the learning with the facility and with the users is really, really important. And we need more training opportunities in open source softwares, and create guidelines or to share those guidelines that are already there. So the organizations like LABI can be an important for gathering these resources. And offers like, for example, in Bioimaging North America is Microscopy DB, which is a database that gathers some information. Is growing and growing. And important is more people helping other people, like giving hours to analyze, to help other people to analyze and to learn to navigate this ecosystem. So at the end, we end up with some specific action. Is what we need is a school at all possible levels, and also to coordinate each others to get awareness with the policymakers and funding bodies, because when we speak about data management infrastructure, we speak also about money, because we need infrastructure, but also we need people. But a really important point that came from Banekal is everything that you do, document it and take the numbers out to see whether you are really creating an impact or not. So thank you for so much. So I'm going to finish my presentation right now.
Thank you very much, Adan, for your presentation. That was very, very nice to see the both presentations side by side and complementing.